A brush the size of a fingertip touches the edge of a 1,000-year-old page. No blade, no solvent, no damage. And inside the dust it lifts off, scientists are finding something nobody expected to survive this long: DNA. Not metaphorical DNA. Actual genetic code, still readable, still sitting inside the skin of an animal that died over a millennium ago, stretched into the very page a scribe once wrote on. 2,000 years earlier, in a cave overlooking the Dead Sea, scribe worked on a different skin, and that DNA survived too. Somewhere in a climate-controlled archive right now, a researcher is running a sample through a sequencer, and the results are starting to rewrite what historians thought a manuscript could tell them. This is not about the words on the page. This is about what's underneath them. This is real science, not speculation, and it has a name: biocodicology. Researchers at North Carolina State University, working with Duke University's David M. Rubenstein Rare Book and Manuscript Library, recently published findings in the journal Manuscript Studies after sampling 91 parchment manuscripts using a technique borrowed directly from medicine. At the University of York, Dr. Sarah Fittiment and Professor Matthew Collins have spent over a decade building an entire field around this idea inside the university's bioarch research group, publishing peer-reviewed work in journals like Heritage Science and Royal Society Open Science. In Israel, an interdisciplinary team led by Professor Oded Rechavi and Professor Noam Mizrahi of Tel Aviv University, working with Professor Matthias Jakobson of Uppsala University, Professor Christopher Mason of Weill Cornell Medicine, and the Israel Antiquities Authority, published a landmark genetic study of the Dead Sea Scrolls in the Bioarchaeology journal cell. And in Germany, a separate team sequenced cattle DNA from 12th century monastic parchments and published their results in scientific reports, comparing the ancient genomes to the modern 1,000 Bull Genomes Project. 4 independent teams, 4 countries, 1 shared discovery. The page is not just a surface for ink, it's a biological archive. To understand why that matters, think about parchment the way you'd think about an old cassette tape. The music on that tape, the text in this case, is what everyone cared about for centuries. But a cassette tape is also physically made of a coated plastic ribbon, and that ribbon itself has a chemical history, a manufacturing origin, a story of where the materials came from. Historians spent hundreds of years listening to the music and ignoring the ribbon. Now scientists have found a way to read the ribbon too, and here's the number that makes this land. The Duke collection alone spans manuscripts written between the late 8th century and the early 1900s. That's over 300 years of unbroken material, roughly the same span of time separating us from Charlemagne's coronation, and roughly 13 times longer than the entire history of the United States. That's not a handful of old books. That's a genetic timeline longer than most civilizations, and the Dead Sea Scrolls push that timeline back even further into skins that were tanned and written on 2,000 years ago, before either Rome or Jerusalem looked the way we picture them today. Here's where it gets specific. Parchment isn't paper. It's treated animal skin, usually sheep, goat, or calf, and every one of those skins carries traces of the living cells it once belonged to. For centuries, that fact was irrelevant to anyone but the animal it came from. Now it's the whole story. The NC State and Duke team, led by researcher Christopher Breen, used a soft cytology brush, the same tool used for a pap smear, gently rubbed across the parchment surface to lift cellular material without ever touching the ink, the binding, or the page's structural integrity. From that dust, they extracted genetic sequences using forensic-grade next-generation sequencing, the same category of tool used in modern crime labs. And what came back mapped almost perfectly onto known regional farming economies. English manuscripts were overwhelmingly sheepskin, about 80% of the authenticated English samples, which lines up exactly with England's centuries-long dominance in wool production. Southern European manuscripts told a completely different story, a much more even mix of goat, sheep, and cattle. Same continent, same century in some cases, completely different genetic fingerprint, because completely different agricultural economies were feeding the industry that made the books. Let's pause here and stack up what we've got so far. A brush, a page, a sequencer, and suddenly historians have a new way to map medieval farming, trade, and craft economies without opening a single archive record. The 12th century English manuscript that received special attention in the NC State and Duke study makes the point especially well. Researchers sampled every single bifolium, every folded sheet, inside that one book, and every page came back as sheet parchment, consistent from cover to cover. Compare that to other medieval books on record, where scribes appear to have deliberately mixed calfskin and sheepskin within the same volume, likely for reasons of cost, availability, or even the texture a particular text called for, and you start to see a level of production detail no catalog entry or library record had ever preserved. That's just one book. But that's just the beginning. Because the next question is obvious. If you can tell what animal a page came from, can that same information solve problems historians have been stuck on for decades? That question is exactly what pulled Oded Rechavi and Noam Mizrahi together at Tel Aviv University. Rechavi studies inheritance in microscopic worms. Mizrahi studies ancient Hebrew literature. They ended up sitting next to each other at a new faculty orientation and realized their two completely unrelated fields might solve the same problem. The Dead Sea Scrolls exist as more than 25,000 disconnected fragments, and for decades, scholars had no reliable way to know which scraps belonged to the same original document. Piece two fragments together wrong, and you can completely change how a passage of ancient scripture reads. So the team, working with samples, sometimes no larger than parchment dust, scraped from the blank underside of a fragment, sequenced the animal DNA preserved in the skins. Almost every fragment they tested came back as sheepskin, with only two coming from cattle. That alone overturned a long-standing assumption that most of the scrolls were written on goat hide, the animal best suited to the dry conditions, around Qumran. But the real breakthrough came next. By comparing mitochondrial and nuclear DNA between fragments, the team could tell whether two pieces came from genetically related sheep, and therefore, likely, the same original scroll, or from unrelated animals that simply looked similar under a microscope. Two fragments long assumed to be part of the same copy of the book of Jeremiah turned out to come from different animals entirely. One sheep, one cow, and the text on them reflected two genuinely different versions of that biblical book. Because cattle could not have been raised in the arid desert around Qumran, that cowhide fragment had to have been carried in from somewhere else. Physical proof, sitting inside the skin itself, that not every scroll found in those caves was actually written there. Now here's the part that connects back to what we saw in England and southern Europe. 7 out of 8 fragments written in a distinctive scribal style that scholars had labeled Qumran scribal practice came from closely related sheep, supporting a theory, argued from handwriting alone for years, that a specific small group of scribes at that site had their own house style. Meanwhile, one fragment of the book of Isaiah carried DNA from a genetically distinct flock, suggesting it was produced somewhere else entirely, and only later brought to Qumran. And a fragment recovered roughly 55 kilometers south at Masada turned out to come from a separate genetic line of sheep entirely, meaning the community there wasn't just reading a copy passed down from Qumran, they had assembled their own, from their own animals. The genetics weren't just confirming guesses. In several cases, they were resolving arguments that pure textual analysis alone had never been able to settle. And biblical scholars outside the project, including Sidney Crawford at the University of Nebraska, have pointed to the sheep lineage findings as real support for a theory that had spent years sitting unproven. Let's take a second recap here, because the through line is worth restating before we go further. In North Carolina, DNA and parchment maps, medieval wool economies. In Israel, DNA in 2,000-year-old scroll fragments is reuniting scattered pieces of scripture and revealing which texts were local and which were imported. Same biological method, wildly different centuries, wildly different questions, and it keeps working. So the obvious next question is how far this goes. Can DNA tell you not just what animal a page came from, but what a document was actually used for by the people who owned it? That's where Sarah Fiddyment's work at York takes over, and it's here that the story stops being about livestock and starts being about people. Fiddyment developed a technique called EZOOMS, adapted from a peptide fingerprinting method to non-destructively test tiny protein and genetic traces embedded in parchment. She used it on a set of 13th century pocket Bibles, pages so thin that historians had assumed for generations they must have come from something unusual, maybe squirrel or rabbit skin, because no cow or goat hide should be able to be shaved that fine. The biomolecular data told a different story. The pages were ordinary calf, goat, and sheepskin after all, nothing exotic. What had actually happened was something more impressive than an unusual animal. Medieval parchment makers had developed a level of craftsmanship so advanced they could shave standard livestock hide down to something almost as thin as tissue paper. Centuries before industrial tools existed to make that easy. The mystery wasn't the animal, it was the hands that prepared it. Then Fiddyment's team went further, and this is the part of the story that tends to surprise people the most. Working with Matthew Collins and a team of collaborators, she analyzed a 15th century object held at the Wellcome Library in London, a medieval birth girdle, a long strip of parchment, inscribed with prayers and worn wrapped around a pregnant woman's body, believed to protect her during labor. Historians had documented these objects for years, but nobody could prove they were actually worn on skin rather than simply owned or displayed. The biomolecular analysis recovered traces of cervicovaginal fluid on the parchment, along with residue from goat's milk, eggs, honey, and several plant species, Ingredients pulled straight from documented medieval childbirth recipes. Fiddyment herself described it as the first direct physical evidence that these girdles were genuinely worn against the body during labor, not just symbolic objects sitting on a shelf. A 500-year-old page had preserved biological proof of an intimate, terrifying, and largely undocumented moment in a woman's life, proof no written record had ever confirmed. In the next part of this, we go straight into what the DNA reveals about deeper questions of authenticity and provenance. Because livestock economies, scroll reunification, and personal use are 3 layers. Verifying a document's true origin is another. The German team working on 12th century monastic parchments extracted enough genetic material from the source cattle to sequence usable genomes and compared them against the modern 1000 bull genomes project, a massive international database originally built to study cattle breeding and disease resistance. That comparison let researchers place the medieval animals within a genetic lineage, giving historians a biological fingerprint that can help verify where and when a document was actually produced, independent of the handwriting, the ink, or the recorded provenance, written by dealers and collectors centuries later. In a field where forged and misattributed manuscripts have fooled experts for generations, a genetic fingerprint from the actual skin the document is written on is not a small development. It's a new category of evidence altogether. And it doesn't stop at the animal. Parchment also holds a microbiome, a mixed community of bacteria, fungi, and trace human DNA left behind by everyone who ever handled, read, stored, or restored that page. Researchers studying ancient Slavonic parchment codices used metagenomic sequencing to map that accumulated biological residue, describing it as something close to a biological pedigree of the object itself, a layered record of custody, environment, and human contact stretching across centuries. Every scribe who copied it, every monk who stored it in a damp scriptorium, every collector who later handled it with bare hands, potentially left a genetic trace behind. The page isn't just recording history in ink, it's been quietly recording the people who touched it the entire time. Here's the part that connects all of this together. Matthew Collins's group at York has taken the science one step further, using a solvent-based technique to extract lipids, fatty compounds, preserved inside old parchment. Those lipids carry oxygen isotopes that shift depending on rainfall and temperature at the time the animal was alive. By analyzing those isotopes across manuscripts from different eras, Collins' team has been able to detect actual historical climate events, including the year without a summer in 1816, the global cooling event triggered by the eruption of Indonesia's Mount Tambora the year before. Collins has suggested that, given enough manuscripts sampled at scale, Parchment could eventually rival tree rings as a climate archive, meaning the same library shelf that holds a medieval prayer book could also be holding a distributed, undiscovered climate record stretching back over 1,000 years. Let's take stock again, because this is the point where the pieces start to form a bigger picture. Livestock economies, craft techniques so advanced they were mistaken for exotic materials, scattered scripture fragments, genetically reunited after decades of guesswork, direct physical proof of how sacred objects were actually used on the human body, genetic fingerprints that can authenticate or challenge a document's origin, a microbiome recording every hand that touched the page, and a hidden climate record buried in fat molecules inside animal skin. None of that was visible in the ink. All of it was sitting there, dormant, waiting for a brush and a sequencer. What's genuinely different right now is the non-destructive part. Older sampling methods for ancient DNA often meant cutting a small physical sample out of the object, something no library holding an original gospel manuscript, a birth girdle from the Wellcome collection, or an irreplaceable Dead Sea Scroll fragment wants to authorize. The Rachavi team, working under strict conditions with the Israel Antiquities Authority, often had to rely on nothing more than dust that had already flaked off the uninscribed underside of a fragment, precisely because cutting was considered too great a risk to even discuss. Breen's team at NC State specifically noted that showing meaningful genetic data can be recovered without damage is what's starting to build institutional trust, opening doors to collections that were previously considered too fragile or too valuable to risk. That trust is why this field is expanding right now, not shrinking. More archives are saying yes, which means more manuscripts are about to reveal what's been hiding in them the entire time. Before this goes any further, it's worth being precise about what this evidence can and can't tell us, because the excitement around biocodicology can get ahead of what the science has actually shown. This is not, in most cases, about testing the DNA of named historical people. When a study identifies the animal a parchment came from, or maps a microbiome, or detects a climate signal from lipids, that's population level and material level evidence. It tells you about livestock, environment, and object history, not about who specifically wrote or handled a page, unless direct biological material from a named individual happens to be preserved and testable, which is genuinely rare. The birth girdle case is one of the few examples where researchers could point to strong physical evidence tied to actual human use. And even there, the identity of the woman who wore it is unknown and very likely unknowable. Even the Dead Sea Scrolls study, for all its power, only tells researchers whether fragments share animal ancestry closely enough to plausibly be the same document. It cannot name the scribe who held the pen. DNA extracted from centuries-old parchment is also badly fragmented, present in extremely small quantities, and vulnerable to modern contamination from anyone who has handled the page since. Fiddyment's team ruled out several sampling tools during early testing, including gecko grip tape, after finding it introduced contaminating cow DNA left over from its own manufacturing process. And Jakobsen, the Uppsala geneticist on the Scrolls project, has been open about how difficult the underlying chemistry is, pointing out that 2,000 years in a cave, followed by decades of handling by researchers not always wearing gloves, leaves a badly degraded and contaminated starting sample to work from. This is careful, early stage, still developing science, not a finished picture, and the researchers involved are candid that funding and further validation are still needed to scale it up. It's also worth saying plainly that none of this rewrites the actual content of history the way a headline might imply. Nobody is claiming these methods change what the book of Jeremiah says, or rewrite the theology of a medieval prayer, or alter the established timeline of when these documents were produced. What changes is context, where a document truly came from, what economy produced it, whether two fragments belong together, and how an object was actually used by the people who owned it. That's a meaningful shift in how historians verify and interpret their sources. It is not a discovery of secret new history hidden in the words themselves. So what does all of this actually add up to? For centuries, a manuscript was treated as a message, words, meaning, argument, prayer, law, trapped in a fixed moment in time. What biocodicology is revealing is that every one of those pages was always a second document running underneath the first, written not in language, but in cells. Written not by a scribe's hand, but by the life of the animal the page came from and every hand that touched it afterward. The ink told historians what people wanted remembered. The DNA is starting to tell them what actually happened, which flock a page came from, which desert cave a scroll never actually saw, which woman's body a strip of parchment was pressed against during the hardest hours of her life. Somewhere in a rare book room right now, a page that has sat quietly on a shelf for 1,000 years or 2,000 is being brushed, sampled, and read for the first time in a language nobody knew it was written in. Tell us in the comments which discovery surprised you more, the medieval birth girdle that preserved proof of how it was actually worn, or the Dead Sea Scroll fragment that turned out to be smuggled in from outside the desert where everyone assumed it was written.